To whatever time zone you're in, good morning, good afternoon, or good evening. The expansion of humankind into space is an international effort, and we are super excited to share with you our experiment design from the little island nation of Singapore. Today, we'll talk you through our idea that could lay the brickwork for building a future in space.
This is our team. We are a trio of 17-year-olds from St. Joseph's Institution International Singapore, united by a common passion for science and space.
Before we start, here's a quote that we feel embodies the spirit of the launch prize competition. Space has the ability to produce a triple bottom line, or ROII, return on investment, innovation, and inspiration.
Our hypothesis is that microgravity conditions will enhance the microbial growth of Sporosaccharina pasteuri and result in a greater cell concentration density by the end of a 20-day period. Now that's a mouthful and quite a lot of information packed into one sentence, so we broke down this presentation into 5 main parts in the bibliography. Let's start off with introduction. What exactly is Sporosaccharina pasteuri, and what makes it so promising?
Sporosaccharina pasteuri, abbreviated as S. pasteuri or SP, is a gram-positive bacteria with a biosafety level of 1, the safest possible classification a microbe can have, so it is perfectly safe to be investigated on the ISS. S. pasteuri is a hardy bacteria that can survive in highly alkaline environments and in the complete and prolonged absence of nutrients, making it a perfect and durable candidate for experimentation in space.
What's really unique about S. pasteuri is its ability to undergo urolysis-driven microbially induced calcite precipitation, otherwise abbreviated as MICP. It takes in urea, a compound found in urine, and then secretes calcium carbonate from it, a key compound that makes up limestone rock. In other words, it turns P into bricks. The mechanisms by which it does this will be explained later on in more detail. In the bottom right-hand corner is the chemical equation illustrating the MICP process.
Lots of progress is being made in the area of producing S. pasteuri bricks, and while they aren't quite ready to be mass-produced at a commercial level yet, researchers are experimenting with different nutrient sources to maximize the strength of precipitated calcium carbonate. The prospects are promising, and it's been scientifically proved that the bricks produced via MICP are of a similar strength to normal bricks.
Now, let's get into the background, importance, and benefits of our project. We started our launch prize journey in April this year, brainstorming the different ways we could tap into the needs of both the space program and humankind. Our initial ideas included things like measuring the decomposition of bioplastics and also the gravitropism of plants. Eventually, we realized that if humans were to become an interstellar species, or simply were to have a greater presence on planets in our solar system like Mars, there would be a need to produce materials in space.
At the moment, there is a hefty price tag associated with shipping things to space. 10,000 USD. That's how much NASA estimates it costs just to ship 1 pound of material to lunar orbit. For space expansion to be economically viable and not drain our resource of cash dry, any production process must have minimal dependence on external resources, meaning that things need to be produced in space, not on Earth and shipped to space.
Now this is where S. pasteuri and its calcite precipitating abilities come in. Urea, a readily available substance found in urine, can be easily accessed to produce construction materials, like biobricks via S. pasteuri. Although the crude image of an astronaut peeing on a petri dish springs to mind and seems like something out of a science fiction novel, it's actually been scientifically proved that biobricks of a similar strength to normal bricks can be made using human urine and S. pasteuri.
Now, if you ask us, that is pretty incredible, and testament to both the resourcefulness of humanity and nature. And we're not the only ones who think that either. Over the past few years, there's been growing interest in MICP in the science community. This bar chart, sourced from the Scopus database on 16 June 2020, shows the growth of publications with keywords microbially induced calcite precipitation, microbial urease, and ureolytic from 1999 to 2020.
Bianca J. Reeksting elegantly articulated our sentiments towards Sporosaccharina pasteuri as the future of industrial production in her March 2020 paper, stating that it represents the paradigm organism for industrial application of calcite precipitation.
Other space organizations, like the Indian Space Research Organization, have explored the possibility of constructing space bricks using lunar soil stimulant containing S. pasteuri. In their conclusion, they stated that their results makes this entire process a very attractive and sustainable option for producing freeform structures using lunar regolith-based building blocks in extraterrestrial habitats.
In an effort to understand more about S. pasteuri and where research currently stands, we took matters into our own hands and emailed Professor Alok Kumar, a key contributor to the paper in the previous slide. And we've been in correspondence with him since June. He's been helping us by answering our questions and sending us copies of his publications. Professor Kumar's 2019 and 2020 research has surrounded S. pasteuri and its possible implementations in microgravity. However, his papers mainly focus on maximizing the compressive strength of S. pasteuri bricks, not investigating the actual behavior of the bacteria in space. In fact, there has yet to be an actual experiment conducted in microgravity conditions for S. pasteuri, despite all the recent craze surrounding it. In order for the dream of producing space bricks to become a reality, we will have to first understand how microgravity actually contributes to the microbial growth of the bacteria in space. And this necessitates the need for an experiment such as ours, a novel investigation that will act as the first step towards making industrial production in space achievable. We will go into further justification for our experiment in the hypothesis justification segment of our presentation.
Calcium carbonate, produced by S. pasteuri, can even help terraform planets. On Earth, research has shown that MICP can strengthen and stabilize soil by filling the gaps between sand grains. In 2020, scientists used the bacteria to improve wind erosion resistance of sand dunes in Iran. If we're looking to eventually build structures on other planets, MICP won't just help us build bricks for infrastructure, but it'll also help us stabilize the physical landscape and ensure that geological features like cliffs and rock faces are secure and safe. S. pasteuri is also a greener and more sustainable alternative to cement, minimizing the damage we could bring to the physical environments of other planets.
So now you see how Sporosarcina pasteuri can benefit the space program, but what about us here on Earth? Sporosarcina pasteuri offers a sustainable and green way of producing materials in general. Traditional brick manufacturing actually creates more pollution than the entire aviation industry, and making a couple of bricks generates more than a kilogram of carbon dioxide. Furthermore, toxic heavy metals and reactive wastes resulting from rapid urbanization can be detoxed by the process of MICP, as it alters the chemical form of these heavy metals from soluble to insoluble, meaning that the toxic elements won't be able to dissolve into our water systems. It is also a relatively new field of interest in the research community, so observing how it behaves in different environments like microgravity could aid our understanding of the organism and help us improve our culturing techniques, move us one step closer to building a better home on Earth.
Naturally, we were super excited about our project and its potential benefits for humankind. This is the exact reason we registered for the contest, to find a way to make a difference. Unfortunately, our microbiology knowledge is limited to high school biology classes, so we had a bit of trouble understanding research papers and didn't really know how to start planning our experiment or what exactly to investigate. We emailed a total of 19 professors from all over the world who have published papers on S. pasteuri. To our pleasant surprise, we weren't outright ignored by all of them, and 8 of them actually replied. To give this presentation without acknowledging their help would be unfair, so we would like to extend our thanks to all the professors who validated our claims and assisted us in this process.
You now have an understanding of S. pasteuri and its incredible MICP capabilities, so hopefully our hypothesis makes a bit more sense. We posit that microgravity conditions will stimulate the microbial growth of S. pasteuri and result in a greater cell concentration density by the end of the 20-day growth period.
Why measure microbial growth and not calcium concentration density directly? This is actually one of the biggest hurdles we faced, as doing so will require laboratory level analysis that is difficult if not outright impossible to perform on the ISS. In overcoming this, we learned that there was actually a direct relationship between the number of cells and its biomineralization capacity. To validate this claim, we emailed 2 professors in the field, Professor Omoregi from Australia and Professor Basaran from Turkey. Both responded positively and provided us with the literature to support the claim of a direct relationship.
This graph illustrates the direct relationship of calcium carbonate production and microbial growth, albeit using the alternative variables shear wave velocity and time. The y-axis shows shear wave velocity, a measure of the mechanical property of soil. As more calcium carbonate is produced, the stiffness of the soil increases, increasing shear velocity as well. The x-axis shows time, which corresponds to the bacterial growth over a specified duration. We can see that the relationship between shear wave velocity and time is directly proportional, illustrating how calcium carbonate precipitation is directly related to cell concentration. This excerpt also offers an explanation to show the direct relationship between calcium carbonate precipitation and cell concentration.
This image shows how S. pasteuri cells produce calcium carbonate precipitates. The cells are actually the location of calcium carbonate production, so increasing the number of cells will logically increase the amount of calcium carbonate. So why do we believe that microgravity will enhance microbial growth? First of all, research shows that motile microorganisms can move into fresh nutrient areas. This means that they can move around in their containers to consume more nutrients. Previous phase experiments have also reported different cell concentrations during early exponential growth periods with respect to ground controls, which gives us all the more reason to believe in this hypothesis.
Professor Rosa Santomartino published a paper recently following her experiment on growth rates of various bacteria on the ISS. She stated in her paper that there have been contradicting results regarding the effect microgravity has on microbial growth. While some professors have found that microgravity enhances microbial growth, others like Professor Santomartino have reported no change whatsoever. In other words, nobody is really sure what effects microgravity has on microbial growth, and researchers like Santomartino have explicitly stated that more investigation is needed, especially with different sorts of bacteria, further justifying our novel experiment with S. pasteuri.
We structured our hypothesis around the 20-day growth period, as this timeframe mimics those of bioindustrial processes, which typically last over a few weeks. We also adapted our compartment design to measure the early growth phase, which lasts about 12 hours of S. pasteuri. So far, there are a few experiments which have investigated early growth phases of bacteria in real microgravity. In fact, most of the research surrounding early growth phases are actually conducted in simulated microgravity. Prof. Santomartino's team focused on stationary phase growth, so they are unable to come up with data for the early growth phase. Other experiments are focused purely on early growth phases, with no data on longer time periods.
One of the first ways we will investigate the bacterial growth is by using real-time optical density measurements. What is optical density? Optical density is a common technique used in microbiology to measure bacterial cultures. It measures the amount of light passing through an optical component, in this case our bacteria-inoculated petri dish. And as the bacteria colonies grow and fill up the petri dish, less and less light will be let through, and our light sensor will record this. Usually, researchers use spectrophotometers to make these measurements, but these machines are way too big to fit into a 100mm by 100mm by 50mm box. So unless you're an AdMan and have the power of quantum physics to shrink objects, you are not going to fit a spectrophotometer on the ISS.
In the word of Bear Grylls, we had to adapt, improvise, and overcome. This next section will detail our compartment design process. Using a computer-aided design software called Onshape, we created a virtual model of our compartment. The compartment contains of 4 individual cartridges.
Inside it, we've placed a small petri dish of diameter 35 mm, supported underneath by 2 cylinders, small cylinders, to prevent the dish from floating in microgravity. Beneath the dish is a sensor from Texas Instruments, which receives light from an aluminium substrate LED mounted on the lid. Here's the LED. Basically, the LED will shine through the petri dish containing a bacteria colony and agar, and the light sensor at the bottom will record the amount of light getting through. As bacteria grows, less light will get through. To ensure that there's an equal distribution of light getting through the petri dish surface, we stuck a diffuser sheet to the bottom of the dish. We came up with a calibration system to regulate the amount of light inside the petri dish. A simple light-dependent resistor, and it's attached to the rim of the petri dish, right around there. And this simple LDR will detect the overall light intensity coming from the LED and any ambient light, and via a calibration circuit it will adjust the internal LED's power to control the whole lighting apparatus. This should account for sudden changes in the light, and it should allow for the total intensity inside the dish and the total intensity incident on the Petri dish to be constant. Furthermore, the whole light apparatus will be wrapped in a black sheet of paper to absorb any stray ambient light, though we expect this to be minimal since the cartridge and lid are made of opaque materials. We have also mounted small 1080p CMOS cameras next to the LED. These photos, along with the optical density data, will be saved to an external hard drive and can be accessed later for qualitative analysis. Everything in our cartridge is powered by batteries. At the moment, we are unsure of the specific model and size of batteries to use, but we did leave sufficient space to slot them inside. This is what all 4 of our small cartridges should look like in one compartment. All the cartridges will be made from a single chunk of aluminium 6061 alloy, as having less individual parts will greatly improve rigidity. This tiny little thing is a vial intended to hold our sample of S. pasteuri if needed. Our cute little Raspberry Pico here is the center of data processing and collection. It is mounted diagonally to optimize space. Here are the extra spaces available for the batteries. This only looks like 4 lids because it is easier to CAD model this way. In reality, these 4 should be one coherent piece, and it should be 3D printed as one piece. This box as well should be 3D printed as one piece. This is again for rigidity and ease of manufacturing. Everything will be secured with screws or glue, and all electronics will be rubber coated to prevent damage in case of a leak.
We came up with the optical density sensor idea prior to discovering that it was an actual technique used by microbiologists, so it was a real relief finding out that our brainstorming had roots in tangible science. It was difficult scaling down the optical density apparatus to the size of our compartment. As far as we were concerned, nobody had really attempted real-time optical density measurement with such size limitations. Nonetheless, we were devoted to our design, as we saw it as one of the best ways to retrieve quantitative data on the bacteria's early growth phase. We created several prototypes of our initial design and emailed the penultimate prototype to Professor Kumar to see what he thought. Fortunately, he responded by saying that he had actually published a paper with a similar design to ours. Honestly, the day he sent this email was one of our year's highlights because it meant that the design we had come up with on our own actually worked. It just so happened that Professor Kumar's design was almost identical to ours, and his publication provided valuable proof that the sensor circuit worked in real life.
Our entire compartment is designed to be easy to use and minimize astronaut interaction. Except for setup and disassembly, it is completely autonomous. Furthermore, the presence of both camera and light sensors ensure that we have backup data retrieval methods should one system fail midway through the experiment. We also plan on taking pre- and post-experiment photos, so in case all else fails, we will at least be able to conduct a cell count to deduce the change in cell concentration density. This wouldn't be a complete travesty, as final cell concentration density does provide valuable results. As Professor Santomartino mentioned in a 2020 paper, more experiments measuring final cell concentration density need to be conducted in microgravity. Overall, our compartment is a 3-in-1 measuring device, allowing us to record data using optical density, camera images, and cell counts. If any one of these techniques are not possible for any reason, the other two serve as viable alternatives to retrieve data.
Here is the basic experiment flow diagram showing the entire process. The pink lilac rectangle corresponds to the flight experiments and the mint rectangle corresponds to the ground controls. Now for the methodology. To understand how bacterial cultures work, we emailed professors from local universities, watched YouTube videos, read methods of similar ISS experiments, researched S. pasteuri culture preparation techniques, and interviewed teachers from our school. We are not experts in the field of microbiology, although I do feel like I could write a paper on S. pasteuri at this point, but we tried our best to come up with a viable method. For microbiology experiments on the ISS, spores of the bacteria first need to be cultivated on Earth. Then they can be frozen and put to sleep, while then transport and eventually woken up by incubating and using nutrient medium on the ISS.
Here are the variables for our experiment. Obviously, the independent variable is the gravitational pull applied on the growth environment of Sporosarcina pasteuri, that is, the 1G on the ground compared to the pseudo 0G on the ISS. The dependent variable, as stated in our hypothesis, will be the microbial growth of Sporosarcina pasteuri. This will be measured by the rate of exponential growth as an increase of optical density as a function of time. The control variables are everything about the growth environment other than gravitational force. This will include the content of nutrient broth, the content of air, the volume of air accessible, the total lux of light incident on the bacteria, total optical density of air between LED and the bacteria, the petri dish and the agar nutrient mixture, the diffuser sheet, the air between the diffuser sheet and the light sensor, all the properties of the electronic devices such as the resistance in the circuit board traces, the time of data collection. This includes the light intensity measurements and photo taking. And lastly, it goes without saying, of course, the bacteria used should be constant, our beloved Sporosarcina pasteuri.
The first main stage of our method is to prepare the bacteria spores suspension. We basically create bacteria pellets to be used as inoculum for the ISS experiment. The second stage is preparing our compartment and samples for launch. We simply need to ensure that all our equipment is in place and working. The final stage will be carried out on the ISS. All the astronauts really have to do is unbox the bacterial samples and wake them up by injecting growth medium and incubating them. After that, the astronauts can leave the bacteria to grow on its own, as our sensors and cameras will record real-time data.
Here's our shopping list of materials. If you ask us, there's no better retail therapy than buying bacteria strains and petri dishes. We can procure Sporosarcina pasteuri from ATCC, and the growth medium actually comes with the bacteria, which is great. We love a buy one get one free bundle. And the sporulation broth medium can be made following the procedure suggested by Monterino et al. In 2005. The hardware equipment for our compartment can be bought from so many different sources, but these are the ones that we suggest. So there's half a year's worth of research. In case it was too difficult to digest all of that, we're going to run through the salient points of our experiment proposal, tackling each of the judging criteria published by the ISET.
Is our experiment feasible? Yes, it is. Similar experiments have been conducted on the ISS before, so it's not a Hail Mary. The Indian Space Research Organization has also recently started to look into designing S. pasteuri experiments, proving that this is a feasible possibility.
Is our experiment novel? Yes. Despite the popularity of S. pasteuri in the scientific community, nobody has actually conducted experiments using it on the ISS yet, which means ours will be the first of its kind. It will be one of the first experiments to measure real-time exponential growth of bacteria in microgravity. It lays the groundwork for humans to become an interstellar species and build habitats on space. Learning more about the bacteria's behavior in different environments can help us understand it better and contribute to developing sustainable production processes here on Earth. And finally, it turns urine into cement, which is pretty darn incredible.
Is our experiment safe? Yes. It has the safest biosafety level of 1, and similar bacteria have been researched on the ISS in the past. Is our experiment automated? Yes. Our custom compartment design, fitted with light sensors and a camera, collect real-time data and save it to an SD card.
Is our hypothesis clear and justified? Yes. We believe that microbial growth will be enhanced in microgravity because the bacteria has the chance to float around in its sediment and move to new areas of nutrient broth. Furthermore, our experiment will provide valuable results into investigating the effects of microgravity on microbial growth, an area that researchers have stressed needs more experimentation. And that's it! Thank you so much for coming and taking the time to listen to us today. We are immensely grateful for the opportunity to be part of this competition, and we went from microbiology zeros to self-proclaimed microbiology heroes. We hope that you enjoyed our presentation and that you will join us on our journey to make biobrick production in space a reality. Thank you again.